Dear students, here Dr. Sivagama Sundari, Assistant Professor, Department of Life Sciences, Kristi Jayanti College. So in this video, it will tell you about the basics of plant tissue culture. So without further delay, we'll move on to the definition of plant tissue culture. What do you mean by plant tissue culture? It is a technique of growing plant cell. So different types of cells are there. Simple tissues, complex tissues. So individual cell you can able to grow and regenerate into a complete plantlet. Tissues, that also it is possible by using plant tissue culture technique. Complete organ and sterile seeds. Seeds are the genetic stock. So here the sterile seeds which has the low germination percentage also can be, by using the plant tissue culture technique, it can be regenerate into a new plantlet in a sterile controlled environment. So that is plant tissue culture. So plant tissue culture, the technique mainly depends on two principles. One is totipotency. It is the nature of the plant, or you can say ability of the plant cell to regenerate into a whole plant. Without that principle, without that one, we cannot able to initiate the plant tissue culture. The next one is plasticity. When you are using the plant cell or tissues or organs or seeds, the plant able to alter the metabolism, growth and development to best suit their environment. Because when you are using the cell, tissues, organs or sterile seeds under controlled environment, it tends to alter their metabolic activity to best suit their environment. So plant tissue culture mainly depends on two principles. One is totipotency, another one is plasticity. So why we depend on the plant tissue culture? So why we are doing this technique? To clone number of plants in a short time. When you check with the normal plant environment in the soil as a supplement, it takes time to mature. Mature in the sense to flower and give you the edible fruits or vegetables, whatever we are aiming for, that edible plant. So to clone number of plants in a short time, we need plant tissue culture technique. And to enable the plantlets to produce desirable traits. Desirable traits is nothing but what the edible thing, the characteristic we want from the plant tissue cultured plant. That is a desirable traits to quickly produce the mature plant. In the sense, if you are using the plant tissue cultured plant, it will flower and give you the edible part very soon within a short lifespan. That does it mean. To multiply plant in absence of seed. No need to rely only on the seed. You can use any part of the plant to multiply into the number by using plant tissue culture technique. Possible to produce plant from seeds have very low chances of germination. Disease-free plants can be quickly multiplied from diseased stock plants. Though you have taken disease-free plant parts from the diseased stock plant, you can able to produce the disease-free plants also. So these are the things we can achieve very short time by using plant tissue culture technique under controlled condition. How the mechanism of plant tissue culture goes on. So adult plant cells are totipotent, meaning they have the ability to give rise to the fully differentiated plant. Because of this nature, it is possible to collect cells from a mature plant and use those cells to produce clones of the plant. Say for example, if you are taking the root of carrot, what happened? The single cell, it has the totipotency, which means it can able to divide. So a clump of undifferentiated mass of the term, we call it as callus, it will multiply. So from the callus, the shoot and root will develop. This is how the mechanism of plant tissue culture usually happens in the technique. Moving on, the two types of culture, based on which type of explant we are using, we can divide into organized culture and unorganized culture. Organized culture in the sense, the culture of whole or parts of the plant. If you are using the complete organ as an explant to initiate the plant tissue culture, we can call it as organized plant tissue culture. Example, axillary bud culture, terminal bud culture, seed culture, embryo culture, ovary and pollen cultures. The second type is unorganized culture, which is not in the form of complete organ. Any proliferation, if you are using that, we call it as unorganized culture. Callus culture, a clump of undifferentiated mass of simple parenchymatous cell called callus. If that you are using it as an explant to initiate the plant tissue culture, we call that as callus culture. And the pieces of callus, we call it as cell suspension culture. Organogenesis, somatic embryogenesis. If you are starting with somatic embryo, then if you are starting with protoplasm devoid of cell wall, a cell devoid of cell wall, we call it as protoplast culture. So moving on to the explants. Explants are nothing but excised plant parts. When you are using the plant parts to initiate the plant tissue culture, that we call it as excised plant parts, we call it as explants. It can be a piece of leaf or a piece of nodal segment, internodal segment, piece of root, or it can be a germ cells from male gametophyte or female gametophyte. Steps in plant tissue culture. So you have to select your explant to initiate the plant tissue culture. As usual, you have to go for surface sterilization, inoculation. So after inoculation, shoot and root emergence will be there. Then you have to subculture. In the subculture, the plant development will take place. Then primary and secondary hardening in the proplates, and finally you have to go for acclimatization. So these are the steps in plant tissue culture involved. So moving on to the factors affecting the result of your plant tissue culture, you have to sure about the genetic makeup of the plant, whether it is haploid condition or diploid or polyploid, those things. Age factor of the plant which you have chosen also matters a lot. Type and source of the explant, from which part you are taking, which type of plant the explant is taken, then nutrient media which you have used. So these are the factors that will affect your plant tissue culture. So with this, we have come to the end of the basics of plant tissue culture. I hope this video enable you to aware about the basics of plant tissue culture, the definition, how the plant tissue culture steps involved, and the plant tissue culture mainly depends upon and the factors affecting the plant tissue culture. Thank you.